Every organism begins as a single cell. So how do we grow, heal cuts, or replace old cells? Well, one disappears or a baby grows into an adult. It isn't magic. It is mitosis. Mitosis is the process of cell division that produces genetically identical cells, allowing the body to grow, repair itself, and stay alive. Before mitosis even starts, the cell undergoes a preparation period known as interphase. During this stage, the cell grows, duplicates its organelles, and most importantly, replicates its DNA. Once everything is ready, the cell enters the mitotic phase, where the actual division of nucleus takes place. This is where the famous sequence known as PMAT begins: prophase, metaphase, anaphase, and telophase. Our first step is prophase. Here, the loose thread-like chromatin coils tightly into visible chromosomes, each consisting of two sister chromatids held together at a centrome. The nuclear membrane begins to break apart, clearing the way for division. At the same time, spindle fibers start to form at opposite sides of the cell, the structure that will later pull chromosomes apart. Next is metaphase. Metaphase is the stage defined by perfect organization.
A stage defined by
spindle fibers
perfect
attached to
organization.
each chromosome,
Spindle fibers
attached
centromere,
to each
guiding them to align
neatly along the cell's equator of the spindle. This arrangement ensures that when the cell divides, each daughter cell receives an identical set of chromosomes. In anaphase, the spindle fiber shorten, pulling the sister chromatids apart to the poles of the cell. Once they separate, each chromatid becomes an individual chromosome. They migrate to opposite ends of the cell, ensuring each future cell receives an identical set. During telophase, the cell begins wrapping up the division process. Two new nuclear membranes slowly form around each cluster of chromosomes. Meanwhile, the chromosomes start to uncoil back into their looser chromatin form. At this point, the cell contains two distinct nuclei, a clear sign that division is almost complete. Lastly, we reach cytokinesis, the separation of the cytoplasm. In animal cells, the membrane pinches inward like a tightening drawstring, eventually separating into two cells. In plant cells, a new structure, the cell plate, forms down the middle, becoming the basis of a new cell wall. By the end of cytokinesis, we have two genetically identical daughter cells, perfectly clones of the original. The process keeps life going, allowing us to grow, heal, and thrive.